All right, welcome to this, well, it's not really short, but not really long either, EdPuzzle on biotechnology. So this is a shortened version of the lecture that I used to give when we would cover this in class. Now it is mostly supplemental material to sort of help you guys figure out, or understand rather, more why you are doing certain procedures in lab and what those things are used for in typical DNA analysis laboratory kinds of settings. So the reason why we do a lot of genetic analysis, one reason, of course, as we talked about in class, is what's known as DNA profiling, right? You're trying to find out where a piece of DNA came from, and so therefore you are collecting what we refer to as genetic markers, and you amplify those genetic markers and then compare the markers in the sample to potential sources to see if you have a match. Another reason to do, essentially to use genetic markers and to perform PCR, right, which is polymerase chain reaction, to amplify those DNA sequences, is to investigate the genes that are associated with diseases in humans. So once we've identified these particular gene sequences, and we have actually sequenced the entire human genome, finally, it took a while because it's big, and through various different types of studies, folks have identified genes that they think are associated with disease. And most of these genes have short, very similar to short tandem repeats, but basically short, what are called single nucleotide polymorphisms. So these are short non-coding sequences that are present on the genome right next to the gene that you're interested in that causes disease, you think. And the presence of certain SNPs, as they're known, can tell you essentially whether you have the version of a gene that is associated with disease. So for example, the allele that codes for Huntington's we know is a dominant allele, and so the SNP that's associated with that allele, they have custom-made primers, PCR primers for specifically that particular SNP. And so the purpose behind, or one of the things behind genetic screening, right, is you take a DNA sample from someone, and you expose that DNA sample to primer sequences that bind to particular SNPs, right, such as the SNP for Huntington's. So if you have it, the primer will bind to that SNP on your DNA sample, and through the process of the heating and cooling and all of that stuff that we do in PCR, it will amplify or make lots and lots of copies, right, of that particular sequence, and then you can separate all the different SNPs using a gel electrophoresis, which I know is something you guys are also working on in lab this week. You can separate those sequences and find out which SNPs you have, and that gives you an idea of which versions of genes that you have, for example, whether you're carrying the allele that codes for Huntington's disease. So those are a couple of different uses of performing polymerase chain reaction, right? Is trying to amplify certain sequences, such as short tandem repeats for the purpose of DNA profiling, or amplifying SNPs for the purpose of doing some sort of genetic screening. Again, we've sequenced the human genome, but aside from us and some bacteria and a few other organisms, we haven't sequenced the entire genome, right? Chromosomes are long, you have thousands of genes, and of course, being eukaryotes, right, we don't know which sequence is to be treated as an intron and which one's to be treated as an exon, so it makes it a little bit complicated. So the most common way we have of amplifying or copying genetic material is polymerase chain reaction. Now, this is a very, very powerful tool. You can take this is revolutionized, as I mentioned in class, revolutionized genetic testing for forensic science, right? You can have a really tiny sample, in some cases the DNA from a single cell, and if you can extract that DNA from the cell, you can use PCR and some just general primers to amplify, most of that sample, and enough, basically make the sample big enough that you can actually do some rather sophisticated genetic testing. So that's the upside of PCR. The downside of PCR is it doesn't handle very long fragments very well, so it does best with shorter DNA sequences. And after, if you've copied something millions and millions of times, you do sometimes get some errors in DNA replication that creep in to that process. So it's fine for most of our purposes in terms of DNA profiling or something like that. However, if what you want is to make the protein product, or you want to test and find out exactly what a gene does, or you're otherwise worried about any errors that creep in, that's a situation where you want to use gene cloning instead. In both cases, though, you're probably going to start off with PCR. Okay, so hopefully folks are at least somewhat familiar, right, with polymerase chain reaction at this point, because I know you've done it in lab. And of course, this is PCR for short, right? And so the whole point of PCR is basically to take a small DNA sample and make it big, right? Make lots and lots of copies so we have enough copies to work with in whatever kinds of analysis we want to do with that particular sequence. So of course, what you're going to do is essentially simulate DNA replication, right? You're missing most of the players in typical DNA replication inside of a cell, right? You're missing most of the enzymes, but you're keeping the really fundamentally important ones, right? You have your DNA sequence, that's your template to be copied. You use cycles of heating and cooling to basically simulate helicase, right? I let the double helix unwind, and when it's unwound, you add primers, right? So in place of primase laying down an RNA primer, you add a primer sequence that binds to a specific sequence of bases on your DNA template, right? And then wherever the primer binds, right, that's the starting point. And then you add your heat stable DNA polymerase. This is your TAC polymerase, right, that we actually got from a prokaryote that was discovered living in the hot springs in Yellowstone National Park. Folks wanted to know, hey, how do these guys function with, at 120 plus degrees, and why doesn't their why don't their enzymes fall apart? And they discovered this heat stable form of this particular DNA polymerase. And again, DNA polymerase will see the available 3' end, and if you add all the available nucleotides, right? So you add your nucleotides, right? You've got your template, you add your primers, and you add TAC polymerase. And then you can allow the DNA polymerase, right, to copy those DNA fragments, right? And it'll just go until it gets to the end of the segment, right? And we'll start wherever the primer starts, right? Now we have general primers that we use for certain sections of human DNA, but if you really want to get fancy and you're amplifying DNA for the purposes of possibly doing gene cloning, often what you'd like to do is actually develop a primer that is specific to the fragment that you want to amplify and nothing else on the genome. And this will allow you can use a gene sequencer to make these custom primers. And if you have a custom primer, that's going to bind, again, to a very specific sequence on the genome and will just amplify that section instead of the whole thing. And this is actually very useful, as we're going to see, if you can do that, for gene cloning. You guys are practicing gene cloning in class, and of course we don't have the resources. We don't have gene sequencers, so we don't have the resources to make custom primers, so we just use sort of the off-the-shelf general primers that we're working with for our particular genes that we're working with in lab. But generally, the idea behind PCR, right, is basically making lots and lots of copies of the whatever gene sequence you have that you're interested in. Right, and so you just do this over and over again, right? I add my template, right, that I have unwound with some heat, right? That opens up the double helix. I added my primer, my Taq polymerase, and my nucleotides, and I let it get copied, right? And then you cool it down, you let the helix re-anneal to itself, right? And that's one cycle. And then you do that again and again and again. You're basically exponentially increasing the number of copies of the DNA that you have, right? And you could end up with, again, millions of copies potentially in this process. Now we know, again, this is great for use with any sort of genetic screening, DNA profiling, a lot of, I don't want to say run-of-the-mill genetic testing, but genetic testing where you're not as worried if you have a couple of changes in the code, right? If you're getting a couple of substitutions or the odd mistake that creeps in, right? Because again, you've got Taq polymerase, the nucleotides, the primer. You don't have any of the spell check polymerases, right? There is nobody checking this product to make sure that it's right, right? And you've copied it a lot of times. So there are going to be some errors that creep in. So if you are worried about this because you want to test exactly what a gene does and you want to make sure that there are no errors in replication, you want lots of copies, but you don't want any mistakes. Or if you actually want to make the protein product that the gene codes for, in either of those cases, then you're probably going to want to perform gene cloning instead of just using PCR. You will use PCR as part of gene cloning, but it plays a much smaller role. Okay, so DNA cloning, as I said, you're going to want to take a gene that you're interested in, and you're going to actually put it in a bacteria and ask the bacteria to copy it for you many hundreds of thousands of times, potentially. And of course, the bacteria have all of those spell check enzymes. So you know you're going to get a faithful copy of the original gene that you inserted. Additionally, if you decide you want to make the protein product, once you've isolated the DNA and all the copies, you can actually ask the bacteria to make that protein product, and you can isolate it and then extract it from there. That's actually how we get human insulin, for example. Coming from your pharmacy, that is produced in what are called bacteria factories. And all those bacteria do all day, every day is make human insulin. And then we isolate it from their colonies and purify it and package it, and off it goes. Because we really have trouble making it on a scale that we can actually manufacture artificially. It's actually faster and cheaper to have the bacteria do it for us. So the principle behind DNA cloning is basically getting whatever gene that you're interested in out of the cells and basically isolating the gene and then putting the gene into a plasmid, which is basically just a circular piece of DNA, right? We know these exist all the time in bacterial cells, and then encouraging a bacterial cell to take up the plasmid, and when it does, right, then you allow those bacteria that have been transformed by absorbing the plasmid, you allow them to grow and copy themselves hundreds and hundreds of times, and every time they copy themselves, right, they are copying the gene that you're interested in, and you're making sure that it's copied correctly, because again, they have all of those spell check enzymes. So the first step in this process, of course, is going to be isolating the DNA that you actually want to replicate. Okay, so what do we do procedure-wise if we want to clone a gene, right? Meaning we're going to use a bacteria to copy the gene that we want for us. Well, we are going to have to start with a little bit of PCR, right? So just like you guys did in lab, you're going to extract the DNA from the cells, right? Isolate whether it's a human cell, frog cell, whatever it is. You're going to extract the DNA, right? And then you're going to copy the gene that you're interested in using PCR, and the primers that you're using are going to have particular what are known as restriction sites, right? So these are basically DNA sequences at the ends that are going to match those that are produced when we are going to cut our DNA with a particular enzyme, right? And so you're going to produce, basically you're going to copy your DNA, your gene that you're interested in. And like we can see on the picture here, let me see if I can get my little doodad to work here. Right, so I have my gene of interest, which is the dark pink part that I've copied, right, with my PCR primer. But then I have these additional ends, right? I've got these light pink bits here on either side. And those are the restriction enzyme sequences, and those are going to match complementary sites when I cut my plasmid with the particular enzyme. And I'm going to be able to stitch those two things together, right? So first thing I'm going to do, again, get my DNA out of my cell. I'm going to copy the gene that I'm interested in. And again, the primer that I use is going to have a particular restriction site at either end, so that no matter which end I'm looking at of my gene sequence, I'm going to have that particular DNA sequence in it that will allow me to cut my DNA with a particular restriction enzyme, right? I'm going to cut my plasmid with the same restriction enzyme, and then I'm going to mix the two together, and you guys, it's exactly what you guys did in lab, right? I'm going to mix together my cut enzyme pieces and my cut plasmid pieces together and add, as we would expect, right, add DNA ligase, right? We're going to add ligase, and ligase, right, is basically going to catalyze that last remaining, right? They're going to bond together, right? The plasmid and the DNA sequence, because they have complementary sequences. So they're going to hybridize together based on those complementary base pairs through hydrogen bonding. But you do need to sort of glue it in place, right, with ligase. That's going to catalyze that last phosphodiester bond on either end of our DNA sequence that we're inserting into the plasmid. Now we have a plasmid, hopefully, we have a plasmid that contains our DNA sequence that we're interested in. We might also, though, have gotten plasmids possibly that just closed back up on themselves and they didn't actually take up the DNA sequence that we're interested in. And because we are not using custom primers and we're not only copying the gene that we're interested in, we might actually have other pieces of DNA that had the same restriction sites as some of the other DNA pieces that we copied in the first place. So we might have to do some cleanup afterwards in terms of isolating our gene after we've done the copying. But for now, we'll just concern ourselves with our transformed, our genetically modified plasmids, right? We've got some recombinant DNA plasmids where I have basically a human gene that I've inserted into this bacterial genome. And then, of course, we do the process of transformation, right, where we encourage, through heat and some chemicals, you encourage the bacteria to take up the recombinant plasmids. And again, they may be taking up plasmids that have DNA sequences that we don't want, and also may have plasmids that are basically blank, right? Now the plasmids, you'll also notice here, we've got a gene that makes bacterial cells resistant to an antibiotic. That's present on the plasmid. So what we typically do is take our right? We've got our recombinant DNA plasmids. We expose our bacteria to those. They take them up, hopefully, right? And then we have a way of getting rid of the bacterial cells that didn't take up the plasmid. We basically plate the colony on a field that has an antibiotic in it, a specific antibiotic, and so therefore only the ones that took up the plasmid and have the gene for antibiotic resistance will survive, right? And so hopefully we'll then get a colony of transformed bacteria. Again, these still may be bacteria that took up a plasmid that had sealed back up on itself and it's basically, blank. Ones that took up the gene that we're interested in, and ones that maybe took up any other fragments of DNA that were produced when we cut our copies of our original PCR products with our restriction enzyme. But that's okay because we can deal with that later. It's not a problem actually. So what the heck is a restriction enzyme anyway? Basically, a restriction enzyme is a enzyme that cuts DNA at very specific sequences. These are the kinds of enzymes that are used by bacteria to protect themselves from viruses. So basically they are, for bacteria anyway, these are enzymes that they use to identify viral DNA and then cut it up into pieces. And if it's cut up in pieces, then it can't hurt you if you're a bacteria. So these are very specific enzymes. Again, they're going to recognize a very specific DNA sequence. These are very short sequences, and wherever the enzymes sort of see that sequence, right, they are going to cut the DNA, right? That means that they're going to hydrolyze the bond, right, that phosphodiester bond between the nucleotides, and basically cut across both of our strands of DNA in order to cut things into pieces. So this is what we call the restriction site. This is where the cut gets made. And the restriction sites are symmetrical. So you have basically the same site. It's a mirror image, right, of itself. So you have one that's in the 5 to 3 direction, and then you have the same sequence flipped around in the opposite direction. And so therefore our enzymes are going to see those sequences on either end and make the cut. And they'll make that cut anywhere that sequence occurs. So our primers that we used, right, are going to have that sequence present, and so that sequence gets copied when we copy our DNA in PCR in that initial step. But again, unless we're using custom primers that are only going to copy the gene that we're interested in and nothing else on the genome, we may get other sections of the genome that were copied when we copied our gene with PCR, and so therefore when we cut our genes with with restriction enzyme, we may get fragments of the gene that we're interested in, but we may also get other fragments. Again, it'll cut anywhere where it sees that particular sequence. And that's going to benefit us because we're also going to cut the plasmid with that enzyme, and that allows us to essentially splice the two things together to make a recombinant DNA plasmid. Okay, so exactly how does a restriction enzyme work, right? It's going to recognize, as I mentioned, a specific site on the DNA, and so anywhere where it sees this particular sequence, it's going to cut, and it's going to cut across both of our sugar phosphate backbones, basically both of our strands. What's interesting, again, the restriction sites are going to be complementary and anti-parallel to each other, right? So same sequence and going in one direction as going in the other direction. So we can see here, right, we've got our 3' to 5' end, and the sequence is CTTAAG. That's the restriction site going that direction. The opposite strand, right, is 3' on the right and 5' on the left. And if you're reading, again, 3 to 5, you're going to still read CTTAAG, right? So the same sequence. Anywhere where it sees this particular sequence, that enzyme is going to make a cut, and it's going to make a cut, in this case, between the G and the A. So it's always making the cut here, and it's making the cut here. And you're going to cut across the sugar phosphate backbone on each template, and that basically breaks it apart. And now you have what are called sticky ends, right? And they're sticky, right? Because we have exposed complementary bases here, right? And they want to pair up with their partner, right? That's all they really want, is to find their complementary bases so that they can hydrogen bond with them. It makes them happy, right? And so what we're doing in our tube with our once we've cut our DNA fragments with our restriction enzyme, we're going to combine them together, and we're going to add DNA ligase, right? If I could get my darn little, hang on, sorry. You're going to add DNA ligase, right? And so what should happen, right, is you've cut your plasmid with the same enzyme that you cut your DNA sequence with, right? And you have complementary, now I have complementary sticky ends. I've got one here, right? And it's going to pair with my sticky end here. And I've got another sticky end on my DNA fragment there, and it's going to pair with my other sticky end there. And they should hydrogen bond with each other, and then again we've added ligase, and so that's going to catalyze our phosphodiester bond here, and here, and here, and here, and that's going to give us our nice recombinant DNA molecule. So that's what should happen, right? But as we mentioned, you could, again, cut the plasmid with a restriction enzyme, and some of your plasmids just close back up, and they don't take up, they don't basically form the bonds, the complementary bonds with our cut DNA fragment. They just seal back up with nothing, right? And again, they could take up the gene that we're interested in, or they could take up any DNA fragment that was cut by our restriction enzyme. Okay, so this little drawing here essentially summarizes our steps of gene cloning, right? So basically what you guys did in lab was get your DNA sample, your cell sample. You extracted the DNA from the cells, right? You then ran PCR, which I'm going to hopefully try and... This is hard to do. It's harder than it looks. Okay. You ran PCR, right? So you copied your DNA using your primers that have that restriction enzyme sequence on them. You're going to copy the gene you're interested in, but you also might copy some other sections of the genome. You cut your gene sequence as well as any other fragments that were cut that had that same restriction enzyme sequence, right? So you may have other fragments that were cut. You're going to cut your enzyme with the same restriction enzyme, and then you're going to mix them all together, right? So your possible outcomes are, I get my plasmids that have the gene that I'm interested in, but I might also get plasmids that have other pieces in them, right? So this one's got a purple piece, and that one's got a green piece. Then, right, you're going to get your bacteria to take up the recombinant plasmids. This is the process of transformation, right? So with heat and some chemical encouragement, you get your bacteria to basically take up our little circular piece of DNA inside them. And you're going to plate them on antibiotics, and that gets rid of any bacteria that did not take up your plasmids, right? But that also means that you may have all these possible options of your recombinant bacteria, right? Ones that took up the plasmid that you're interested in, that has the gene you're interested in, excuse me, and then ones that took up our other plasmids that have other sequences that we're not interested in, right? Plus the ones that took up a blank, right? So ones that took up an empty plasmid, which I'm just going to call blanks, right? Ah, it's like a kid's writing it. Okay. Right, so these are your bacteria that took up the plasmid, but the plasmid had just closed back up on itself, right, and it doesn't have the gene in it. So the next step you're going to do is let your bacteria grow and copy themselves like crazy, right? So you're going to get lots of blanks, you're going to get lots of ones that have your gene that you're interested in it, and you're going to get lots of ones that have any other DNA fragments in there. But you're going to get all transformed bacteria because you got rid of the ones, right, that didn't take up the plasmid because you're growing your bacteria, right, in a field of antibiotics. And the plasmid contains the gene that allows them to resist the antibiotic. So you're only going to have colonies of transformed bacteria, but you won't necessarily know which ones have the gene that you're interested in. And that's actually okay. We can actually deal with that problem. It's not actually a big deal, and I will show you why. Okay, so now we've got our colonies, right, of our bacteria, and we've grown them a whole bunch, right, incubated them, so they've basically gone through a whole bunch of rounds of binary fission, right? They're basically copying themselves a whole bunch of times. So now we have copies of all of those DNA fragments, including the one that we're interested in. They're just in the bacteria. So what we have to do is essentially what we did at the very beginning, which is extract the DNA from our bacterial cells. And you guys are going to do this in lab this week. You're going to extract the DNA from your bacterial cells, and then you're going to separate. Basically, now you have a suspension of all of your DNA fragments from all of your bacterial cells. And then what you're going to do is put that mixture into a agar gel, and then you're going to apply some electrical current to that. So be careful because the voltage is no joke involved in this. So be careful. Follow your TA's instructions very, very, very carefully. So what you're putting in initially, right, is going to be a mix of all of your DNA fragments, but after you've applied the electrical current, the DNA is going to move through the gel, and it's going to move a particular distance based on the charge of the DNA itself, right? So DNA overall is slightly negatively charged, all those phosphates, right? And so it's going to be attracted to the positive pole. So it's going to travel that distance, but right, you have different sequences of different lengths, right? So the longer sequences are going to be heavier, and they're not going to travel as far up the gel as shorter fragments. So assuming that we have an idea of how big our gene of interest is, we can figure out which one is our gene of interest on our gel. And then what we can actually do is, using a different type of tool, but similar to our pipette, is essentially extract the DNA from the gel once we've separated it. Then we know it's just the gene that we're interested in, and that's it. We basically have purified our DNA sample. Dun, da, da, da. And now I have a bunch of copies of the gene that I'm interested in. And the question, of course, then becomes, well, now what do I do with it? What can I do with that? I've got a bunch of copies of a gene. And the answer is, well, what can't you do with it, right? You can take all of your copies of your gene and you can put them in another organism, right? Quite often we put them in a bacteria, or you put them in, you can actually put them in a chloroplast in a plant, right? You put them in animals, you can put them in other places and make genetically modified organisms that now carry genes from other creatures, right? So you can certainly do that. You can take all of your copies of your gene and you can do other kinds of analysis with it, right? You can sequence the gene. You can do other kinds of things with it, or you can you can essentially clone it again, in a sense, and you can repeat this whole process with just your gene fragment, right? You still have to get rid of the untransformed bacteria, and you'd have to have a way of getting rid of your bacteria that took up the blank plasmids. Or in some cases, you might not care about those, right? If you have a small enough number, you actually don't care about those. But you can essentially transform the bacteria again, and in this case, actually allow the bacteria to make whatever that gene codes for. Haha, right? And so this is, again, a lot of the field of proteomics is finding out, hey, what does this gene code for? What does this gene do? Let's make the protein product and find out what its shape is like and what exactly it is, and that gives us an idea of what it's for, right? Because form and structure and function go hand in hand. We've learned that pretty solidly by now, right? And so again, you can make the proteins, you can express those new traits, right? Whatever those traits may be. And so like I mentioned earlier, right? Human insulin. We put the gene for human insulin, we stuck it in a bacteria, and now the bacteria basically are just cranking away, expressing that gene, and they're making that protein product. And all we have to do is find a way, we have a particular protocol for separating the insulin from everything else in the bacterial culture environment, and then you can purify it and you're done. Da, 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 da. And I just keep having my bacteria make that stuff over and over. They can make different kinds of proteins to, for example, dissolve blood clots in heart attacks. This is called TPA, and we're going to get into that more when we talk about the circulatory system, but you can essentially have it make the protein product if you're interested in that. Now that's really your only option. If you want to make the protein product, you really need to have that gene inside a bacterial cell, right? Because if you're in a eukaryote cell, right, you may not get the exact same protein because of all those introns and exons, right? We do all that alternative RNA splicing. And if you're just doing straight up PCR, you can't get the protein product either, right? So the only way you can do that is if you actually essentially make a recombinant plasmid and transform the bacteria and let the bacteria make the protein product. Okay, so I've got a little video here put together by some grad students from MIT, basically doing a short overview of the steps of gene cloning, in this case using some examples of DNA that they got from a frog.
Hey there! You may be wondering what you're doing at the front of a bioengineering lab and why a total stranger is talking to you right now. But I'm here to talk about some of the awesome experiments that Herbert Boyer and Stanley Cohen did in the 1970s. You might be thinking to yourself, the 70s? Isn't that like when the dinosaurs lived? Boring. Well, have you ever gotten a vaccine or eaten groceries from a grocery store? That's all thanks to some of the experiments that these two lab pals did in the 70s, where they took enzymes that cut DNA and were able to insert these pieces into new bacterial DNA. They pioneered the field of genetic engineering, and thanks to all the work that they did, we have medications for people with diabetes and heart attack victims. You know what, why don't you guys just follow me inside? So the first thing that Cohen and Boyer did was they took DNA from a frog and cut it up into pieces. I should probably explain to you guys what's going on in this tube. Here's some DNA from the frog. It encodes certain traits that affect how the frog develops, how it survives, how it looks. Now there's a piece of DNA in this genome whose job is to encode for certain proteins that the frog uses to survive. We're going to cut this piece out using a restriction enzyme. This is the thing that Boyer discovered. It's kind of like a pair of scissors. By cutting the DNA at specific points, we can get different pieces. Now, there are tons of different kinds of restriction enzymes, and they all cut in different places in DNA. Our enzyme that we're using today is called EcoR1. Now, this piece of DNA has more than one spot that EcoR1 can cut, as shown by the dotted lines. So we're going to end up with other pieces of DNA in addition to the one we actually want. Now, that's actually okay because we're going to deal with that problem later. What's going on in the tube is we're literally just cutting up our piece of DNA just like this. Now, here's a tube full of plasmid. It's basically a circular ring of DNA that can exist in bacteria, and it basically encodes for certain traits that affect how bacteria look or act or survive. Now, to add our DNA from the frog DNA to our bacterial DNA, we need to cut the plasmid open too. So we've got to add our Echo R1 scissors, and they're going to cut at this site. You let this reaction proceed, letting all the ingredients, the plasmids, the restriction enzyme, and some water and buffers to hang out together until all the plasmids are cut like this. Now we need to add the DNA pieces from the frog and allow the pieces to join our cut plasmids. Here's what's going on in the tube now. I've got pieces of frog DNA, and I've got cut plasmids like this. I need to add this guy into this plasmid. And the way I'm going to do that is by adding an enzyme called DNA ligase. This enzyme kind of acts like tape, and it fixes the DNA insert into the plasmid. Now, another important thing about the plasmid itself is that it carries a marker that makes it resistant to a certain kind of antibiotic called tetracycline. This will come in handy in a little bit. After a little while, we'll have a couple types of DNA going on. This is the one that we want. It's plasmid and it has the purple frog DNA insert. But we'll also get things like this. We'll get plasmids that had other parts of the frog's genome inserted into it. And we'll also get plasmids that just close back up on themselves. So now that we have these new plasmids, we've got to stick them into some bacteria. Here we're going to take some E. coli. As you can see, it already has some DNA in it. The important thing about these guys is that they'll actually die if you add the tetracycline antibiotic, which we talked about earlier, to them. No. What Boyer and Cohen did to get the plasmids inside of these E. coli is that they cooled all the cells down and then quickly heated them up, creating little holes in the cell membrane. Then the plasmids could slip through and get into the cell. These are the kind of bacteria that we have now. Bacteria that got the plasmid we wanted. These are the guys we want. But we'll also get bacteria that got the plasmids that we didn't want, say this one with the wrong piece of DNA, or this one with just a plain plasmid without any frog DNA. And more than likely we'll get ones that just didn't pick up any of the plasmids. Now what I'm going to do next is plate these cells on some tetracycline agar plates, which is basically some fancy jello that has antibiotic in it. So these guys won't be able to grow, but these guys will. I've plated the E. coli that we transformed with our new plasmids onto this plate, which has tetracycline in it. We're going to let this sit in the incubator overnight and allow our bacteria to grow, and we'll check on it again in the morning. Alright, so as you can tell, we've definitely got some colonies that grew on our plate overnight. So now the problem that we have is we need to be able to distinguish the bacteria that got these plasmids from the ones that got these plasmids, which don't have our frog DNA in them. So what we're going to do is use the same techniques that we did at the very beginning of this experiment and extract the plasmids from all these bacteria. Then we're going to use the same restriction enzymes and cut up these plasmids. You might be able to notice that our frog purple insert is a different size than the pink that comes from the background frog DNA. So what we can use is something called a gel box and use a technique called gel electrophoresis. And what it's going to do is it's going to separate our different pieces of DNA out. And from this experiment, we'll be able to tell the plasmids that took up the frog DNA from the ones that didn't, or from the ones that just closed up on themselves. Thanks to these cloning techniques that me and my buddy developed, the field of genetic engineering was pioneered. Things like insulin are being made these days, using these very same techniques. Well, that's all I've got time for today, guys. Thanks for stopping by. Hopefully you have a better appreciation for some of the cloning techniques you saw today and just have a better understanding of genetic engineering. See you guys later.